is found in snapdragon plant. Sample or Snapdragon plant. Generally, the Snapdragon plant flower types red, white. Now, parents choosing true breeding red flower Snapdragon plant. This is actually capital R expressed as capital R. Second, true breeding white flowered Snapdragon plant. Homozygous, smaller. So these two best students, if you go to the white flowered Snapdragon plant. Just a minute. Now, this is the parent chosen. So it is homozygous. R for red and small r indicating white. Now you see, now about the cross. I'm rubbing it out. Can I rub it out now?
Sir, please wait. Sir, what it is written? Flower.
Huh? Red
Flower.
flowers. Which one? Flower types. Flower types. Completed?
Yes, sir.
Can I rub it out?
Yes, sir.
The P generation. Red. Crossbreeding. Gametes. Cell breeding. So this is what about the incomplete dominance. You see where it is found. Incomplete dominance doesn't follow Mendel's law of dominance. You can write here. Mendel's law of dominance. But it follows
স্যার উপরে একটা ফলো হবে।
wow. Law of segregation. Remember this thing. Now, here hello. 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 So this is the another most important observation that in case of incomplete dominance, genotypic and phenotypic ratio will remain same. Most important thing you need to remember here, you can write it down also. In case of incomplete dominance, the genotypic ratio and phenotypic ratio are found same. Clear? You please write it down. In case of incomplete dominance, the genotypic and phenotypic ratio are found same. Written?
Yes, sir.
So this is the most important thing here. What does it mean, the incomplete dominance? You know, capital R, and smaller, the two parents. Whenever R and R are stay together, the colors of the flower is found pink, neither red nor white. So you can say here that one out of two alleles in a allelic pair, one is not going to dominate the other, right? In this case, in a dissimilar pair of alleles, one factor or allele is not found to dominate the other. That's why the term incomplete dominance is used. Why? How can you say that dominates the other, cannot dominating the other? How can you say that it is not going to dominate the other? Because if red can dominate white, so whenever capital R and small r present together, according to Mendel's law of dominance, R to dominate the small r so that the flowers become red, but it is not happening here. That's why the term comes incomplete dominance. Now, after that, we have to proceed towards the concept of dominance. Is it clear to you or any doubt?
Clear, sir.
In this finding, R is not so the thing happens here, R is not completely dominant over the smaller. So I am going to rub it out now. Can I rub it out now?
Yes, sir.
Okay, thank you. Concept of dominance. Next one is concept of dominance. Now, what about it? You see two cases. Capital T, small t, capital T. Go to these two examples. Then how it is happening actually? It is said.
স্যার এটা ক্যাপিটাল টি স্মল টি হবে না?
কোনটা জানি? Oh, sorry. Now it is said in a heterozygous capital T and small t, they are found slightly different from each other, as it is said that two alleles are slightly different from the same gene. And you know, if we go through this, what about them? You see, allele T allele T, two different alleles. So what happens? Allele T can produce a normal enzyme which is responsible for the controlling tallness of the plant. Now, what about the small t? How is small t developed? Actually, capital T is the wild type. Present from the beginning. Now, in the course of time, in some places, when short or dwarf pea plants are required to be developed, as per the need of the environment, capital T undergoes mutation. Capital T gene undergoes mutation to form small t gene. Are you getting me? Suppose initially, only the first evolved plants are all tall pea plants, this is capital T. Later on, in order to adapt in different environmental conditions, slight modification of capital T is required for ensuring survival of the pea plant in a particular habitat. Then this capital T gene undergoes mutation, and its mutated form is the small t. Is it clear to you? Is it clear?
Yes, sir.
Now this mutated form, whenever present together, then dwarf plants are produced. Like this, whenever you will get small t together, you will get a dwarf pea plant. Now, what happening here is, so the mutant type allele so here we can write here capital T mutation small t. So from this we can write capital T is a normal allele, small t is a modified allele. Capital T is a normal allele and small t is a modified allele. Modified, and it is modified due to the mutation. Now, what about this modified allele? Look. The modified allele, here also, here what happens? Capital R is there. And modified allele. So the modified allele is responsible for the production of what? Normal so although it undergoes mutation, so but it can produce a normal enzyme or non-functional enzyme. No enzyme. First thing I should say you, suppose one allele, capital T. So this is the capital T gene.
স্যার লেফট সাইডটা দেখা যায় না।
Oh. Capital T gene, allele. Okay. Okay. Capital T allele. Just 1 minute, please. T allele. Transcription, mRNA, translation, protein, enzyme. Transformation of substrate. In this case, C plant becomes stall. Back. Is it clear? T allele undergoes transcription, mRNA undergoes translation, protein form enzyme, enzyme acts on substrate, leading to the transformation of substrate, and P plant becomes stall. Are you getting me now? Clear? On the other hand, look, small t allele, oh. Small t allele in homozygous form. In homozygous form. small t allele in homozygous form. Just 1 minute, please. Okay. Please.
Kita go. Kita go. Sorry, Sajain. Makbuk tayo boy kasi ko.
Just 1 minute, please. Okay. Now, see here, allele in homozygous form, transcription. mRNA translation. थोड़ा you can write is non-functional enzyme. Or it may not even undergo transcription. Triplen becomes dwarf. It is one sense, it may happen like this. So that in this way, triplets become dwarf in homozygous condition. Is it clear to you?
স্যার ট্রান্সলেশনের নিচে কি কিছু এঁকেছো না কেটেছো?
এটা হচ্ছে non-functional enzyme. Non-functional enzyme.
আচ্ছা আচ্ছা
প্রোটিন একটা স্টেপ মিস করে যায়। Clear now? Is it clear? Now I have to now what is shown here in basically in homozygous condition. Capital T, capital T. You please note it down. It is a homozygous in homozygous form. It is in small t in homozygous form. It is shown. Now we have to proceed towards the heterozygous condition. Clear? Now you see this is most important. Now from here you see in the first case that is in case of A, as you produce normal or less efficient enzyme, so we can say the modified allele equivalent to the normal allele. So in this case, the modified allele will produce similar phenotypic as it is producing the normal or less efficient enzyme. I'm talking about this one. As it is normal and less efficient enzyme, so it is equivalent to the normal allele. That's why in capital T small t combination, in capital T small t combination, the P plan is found tall as the modified allele is equivalent to normal allele. Is it clear now? Look at this. In the first case of A, there is a modified allele is equivalent to the normal allele. So, in this case, the modified allele will produce similar phenotypic effect. That's why in TT combination, in the P combination, the P plane is found tall as the modified allele is equivalent to normal allele. So its phenotype is not affected when a modified allele is found along with the normal allele. Clear? Because here, whenever a modified allele is found along with the normal allele, the modified allele is going to produce the same phenotypic effect as it produces either normal or less efficient enzyme. Clear? Is it clear to you?
Yes, sir.
Now, in case of not to write. Okay. In case of RR allelic pair, found in Snapdragon plant. In case of RR allelic pair found in Snapdragon plant, the R allele is the modified allele. Produces a non-functional enzyme or these allele produce no enzyme. So in case of RR allelic pair found in Snapdragon plant, R allele produces a non-functional enzyme or this allele produces no enzyme. Hence, phenotype is affected in this case. Clear? Is it clear now?
Yes, sir.
Yes, sir.
So the unmodified allele is actually the dominant allele. Or you can say right. Can I rub it out now? Can I rub it out, please?
Yes, sir.
Yes, sir.
Yes, sir.
So unmodified allele or normal allele. Suppose T is it dominant allele. Whereas, on the other hand, the modified allele small t allele P plant stem height character is a this is evident. That's why any recessive trait already I shown you in case of dwarf P plane, any recessive trait is found due to the non-functional enzyme or no enzyme produced by the specific allele. Are you getting me? Is it clear to you? Is it clear?
Yes, sir.
You can go through your book also. The concept of dominance, you can go through it. I'm giving you 2 minutes time, please. Go through it now, page number. What is the page number? Okay, after incomplete, you will get it. Please open the book. Do you have the book with you? Do you have the book with you?
Yes,
Yes,
sir.
sir.
Okay, open it up, please. And just read out this passage, please. Concept of dominance. Quickly, I'm giving you 2 minutes time or 3 minutes time, please. It's very much required now. Go through it.
স্যার
Yes.
স্যার আমার একটা কোয়েশ্চেন ছিল
বলো।
স্যার বলতে এখানে এখানে তো তুমি বলেছো যে মানে মানে মডিফাইড এলিলটা ইকুইভ্যালেন্ট হয় নর্মাল এলিলের জন্য ওই ক্যাপিটাল টি স্মল টি কম্বিনেশনে, তাহলে মানে স্মল টির ক্ষেত্রে এটা ডট কিভাবে হয় এটা আবার একটু বুঝাবে।
Actually, capital T can express itself in the presence of small t. A small t can be expressed in the presence of capital T. But whenever it is in our recessive form, when a homozygous recessive form, it cannot express itself. So here, it is triggered by the effect of the capital T. Are you getting me?
ইয়েস স্যার।
Okay. Now, in the case of capital T, small t, sometimes it may also be happen that capital T express itself, small t is not required, so that it becomes tall. But in case of incomplete dominance, that's where some problems occur. It may also happen like this also. Is it clear to you? Can I rub it out now? Can I rub it out now?
Yes, sir.
So remember this thing here, there's something, sometimes it is happening differently also. In case of capital T, small t, capital T is dominant, small t is not producing, although then it becomes capital T. Sometimes actually the capital R and small r are present together, R is required to become active to facilitate expression of capital R completely. So this is also happening here. So remember this thing, in case of concept of dominance, one thing you need to remember, what I showed earlier, the capital T gene is to produce the normal enzyme. Sometimes in some cases, the what is called the distributor also can produce the normal enzyme, already it is shown to you. Now, then comes to the codominance and multiple allelism. Codominance and multiple allelism. What happens in case of codominance, you see, when two dominant alleles located on the same gene locus, when two dominant alleles located on the same gene locus, express themselves independently, then it is called codominance. Then you can call it codominance. Another term is multiple allelism. When more than two alleles located on the same gene locus, responsible for the expression of a are responsible particular phenotype, then it is called multiple allelism. Example, ABO blood grouping system. In case of ABO blood grouping system, you will get this. Suppose I am showing you. In case of the example is look, but you can write it. Actually, blood group is controlled by three alleles, namely IA, IB, and I. Clear?
স্যার আই এ কেন?
Basically, there are three alleles. Some alleles like producing particular sugar. IB produces a different type of sugar, and I cannot produce any sugar. I allele can produce a particular type of sugar. IB allele can produce a different type of sugar. Clear? I allele can't produce any sugar. Is it clear to you now? IA, IB, and I are the different forms of one of the gene I. So iodine found to be in the IA form, IB form, or I form. Now, how can they be arranged? Here, IA and IB are dominant alleles. I is a recessive allele. So, can I wrap this? Can I wrap this part?
Yes, sir.
Now you see, I and I. Is it clear? In this case, oh, এটা কি দিছে? ঠিক ঠিক দাও তো। জানলে দাদা কি 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 করতো? কি? এটা? কি দেবে? রাখছি কেন আমি? দিছি দিছি। Now, look at this example. In case of IA and IB, so you see, IA, both are dominant blood group is A. As IA domination was small I, it should be A. B, it should be blood group B, AB. And in case of AB, in this case, express themselves independently, and hence a different blood group is formed. Called AB. IA and RB express themselves independently and hence a different blood group is formed called AB. Clear?
Yes, sir.
And this is codominance actually. And it is called codominance. Clear? Is it clear? Now, if you go to the, can I rub it out? Can I rub it out?
Sir.
Completed?
Yes, sir.
Okay. Look one thing. This is one parent. You see. IA high B. These were actually allele from clear. And it's very important for the board and also for the entrance. So number of combination here should be 6 different combinations of form because the number of alleles is 3. When number of allele 2, then there are 4 different combinations. When number of alleles is 3, there should be 6 different combinations. Is it clear to everybody? Is it clear?
Yes, sir.
Okay. After that, sometimes it is found that in some cases, they will get a mixture of dominance and incomplete dominance together sometimes. while sometimes two seeds are round and wrinkle outwardly, but inwardly they have some three characters. Actually, capital B for round seed, small b for the wrinkle seed. But in the phenotype, capital B round. Suppose, can I rub it out? Can I rub it out?
Yes, sir.
Yes, sir.
Sometimes you see, look, there are two alleles, suppose one is small b. Two alleles. Remember this thing? It's called capital B, small b. So you see, whenever it is capital B, and small b, it is crossed. Capital B, small b, capital B, small b. Phenotypically, the round shape, the wrinkle shape. So it is found to be round. Again, if it is crossed, capital B, capital B, small b, capital B, small b, small b, small b. Clear? Here it is round, 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 and wrinkled. Clear? But in terms of size of the starch grain, you will get three sizes. Large size, starch grain, and small size starch grain. Clear? So, if the size of the starch grain is concerned, there is incomplete dominance here, right? If you consider the size of starch grain, considered, it is a case of incomplete dominance. But if the shape of seed is considered, then it is a case of complete dominance. Clear? Is it clear to you? Here you will get the case of both complete and incomplete dominance together. Considering the shape of the sheet, it is complete dominance here. Consider the size of the starch gain, it is in case of incomplete dominance. So you are getting both complete and incomplete dominance together in the same example. Clear?
Yes, sir.
স্যার এটা কি পি প্ল্যান্টের ক্ষেত্রেই হয়?
No, no, P plant. No. This is the common example from any plant. A single gene product producing more than one effector gene. Yes, single gene product is responsible for more than one effect. One effect is size, shape becomes round or wrinkle, other is the size of the starch gain. Is it clear?
Clear, sir.
Okay, thank you. Now today I am going to conclude here. The next class I am to start with the inheritance of two genes, right? You go through these today's topics. If you have any doubt, we'll be discussing it again on the next day. Okay?
Okay, sir.
Thank you. I am concluding now, okay? Note it down, everything?
Yes, sir.
Okay, thank you very much.